Excitation is the process by which a motor neuron's impulse stimulates an action potential in a skeletal muscle. It is essentially a chemical synapse between a neuron and a muscle fiber, meaning it utilizes chemicals called neurotransmitters. Since this specific type of synapse is between a neuron and a muscle, it's called a neuromuscular junction. It's also known as a motor end plate. We can look at excitation as a step-by-step process. It begins with the nerve signal in the motor neuron reaching the synaptic knob and stimulating the opening of voltage-gated calcium ion channels. Since calcium ions are higher in concentration outside the cell, they diffuse into the synaptic knob. The calcium ions trigger the exocytosis of vesicles containing a neurotransmitter called acetylcholine. This is released into the extracellular fluid between the neuron and the muscle fiber. This space is called the synaptic cleft. Diffusing across the cleft, acetylcholine binds to receptors on the sarcolemma, which is the muscle cell's membrane. The receptors are ligand-gated ion channels, and when they open, sodium ions diffuse in and potassium ions diffuse out. The sarcolemma rapidly depolarizes and then rapidly repolarizes in an event called an end plate potential. The end plate potential stimulates adjacent voltage-gated sodium and potassium ion channels in the sarcolemma to open. These ions diffuse down their concentration gradients, depolarizing the sarcolemma. This result is called a muscle action potential, and the muscle is now excited.